Hi, I'm Amatur Rahman. I'm a fourth year PhD student at Penn State University working with Dr. Paul Medvedev, and today I will present our work where we uncover two hidden assembly artifacts. So reconstructing the sequence of a genome from its sequencing data is a very challenging task in bioinformatics, and despite many recent advances, assembly errors still remain a major limitation. So assemblers suffer from these two types of errors. One of them is misassemblies, where you put together sequences that should not be adjacent, and the other is underassemblies, where you do not put together sequences that should be together. And there are some ways to reduce these errors, like long-range technology, long-range contact mapping, manual curation, and combination of these methods are being used by recent assembly projects like T2T, VGP. However, these approaches remain time and cost prohibitive for most labs. So we kind of want to find the algorithmic sources of these errors. So how do we do that? We decided to investigate by looking at the common denominator of many assembly algorithms, which is Unitig algorithm in the Brown graph. So how is a de Bruijn graph constructed and what's a Unitig algorithm? So in assembly problem, you're trying to reconstruct this sequence from the set of reads given to you. Here, the reads are of length 4. And to do that, you first build the set of k-mers from it. Let's say k value is 3. And then those k-mers are the vertices in the graph. And you put edge from k-mer x to k-mer y if the suffix of length k minus 1 of x is equal to the prefix of length k minus 1 of y. And that way you build a de Bruijn graph. Now, to get the Unitigs, you find all the maximal non-branching path in the de Bruijn graph. And in this way, you get these 4 paths, and these are the 4 maximal Unitigs. And the corresponding strings are AC, TTG, TSC, TGG, and TGSC. Note that all of these 4 strings are substring of the sequence. So we say that all of these 4 are safe Unitigs. Now the assemblers expect the safe Unitigs when there's error-free reads. And so the most common misconception is that Unitigs are safe in case of error-free sequencing. But as we will see in this work, that's not exactly true. Okay, then there's the other thing that these Unitig algorithms need to handle. It's a DNA double-stranded nature. So when you have these reads, you do not always know from which DNA strand it's coming from. So to handle that, assemblers use this 2 different kind of approach. So on the left, there's a double-directed de Bruijn graph where the input k-mer set is doubled. You put both a k-mer and its reverse complement prior to constructing the de Bruijn graph. And in the bidirected de Bruijn graph, you don't double the k-mers. Instead, you use these bidirected edges, which have 2 orientations, okay? So because this bidirected de Bruijn graph has this elegant construction, it's quite popular, and the expectation is that bidirected graph should not hurt assembly accuracy. But as we will see, that this is not true. So that's our contribution, that's our finding in this paper. We uncovered these 2 sources of error that we show that unsafe Unitigs cause misassembly, and in bidirected de Bruijn graph, the bidirected edges break palindromic Unitigs in half, causing underassembly. And we uncovered these errors using theory and confirmed them using experiments. So let's start the first part of the study, which is about Unitig safety. So if there are sequencing errors, that will make the Unitigs unsafe because the reads themselves are not going to be exact substring of the sequence. What about in error-free data? If coverage is perfect, Unitigs are safe except in corner cases. This was proved in an earlier work. If coverage is not perfect, it is unknown how the safety is affected. And previous work suggested that they may be unsafe. So our goal is to find non-contrived conditions for unsafe Unitigs on error-free data when coverage is imperfect. So this is the example we saw earlier, where all the Unitigs are safe. And now we move on to this imperfect coverage case where this KMR, GAC, and TG is not sampled by the reads. So we end up with the Unitig TSC TTGG, which is not a substring of the sequence. So to capture the notion of safety formally, we introduce this concept of sequence segment, and we say that a Unitig is safe if it is a substring of sequence segment. So what's the sequence segment? Here in this figure, we marked three sequence segments in three different colored rectangular boxes. And these are basically strings corresponding to a maximal sequence interval. And these sequence segments do not overlap by more than k-2 bases. So these three purple reads here, they constitute this interval or this sequence segment, and this pink and this green sequence segment, they do not overlap by more than k-2 bases. It just has an overlap of 1. Okay, so now that we know what sequence segment is, we can start with a set of sequence segment, and then we can answer whether a particular Unitig W is unsafe. And we can do that using this theorem that we've proved in our paper. And this is the main theorem for unsafe Unitigs for imperfect coverage. Okay, so in the figure, for each of the sequence segment, we have a walk corresponding to it in the same color. And I'd like to walk you through the 4 cases or the 4 conditions of unsafe of Unitig being unsafe, one by one. So first of all, this TGSC segment, it does not contain any k-mer of the Unitig in question. So this is the Unitig, right? SCTCTTTG. Let's call it W. And no k-mer of it is in TGSC. So the first condition holds for TGSC. And for the second condition, it says that this segment starts somewhere in the middle of the Unitig, and that start occurs exactly once. The third condition is kind of similar. Instead of start, it's just the ending vertex that occurs only once, and the end is somewhere in the middle of the Unitig. And for the fourth condition, we see that the segment starts somewhere in the middle and somewhere in the middle of the Unitig, and the starting k-mer and the ending k-mer occur exactly twice in the segment. So as we see that the all 4 segments in this graph, they do not contain W, and as a result, W is an unsafe Unitig for this example. So that brings us to the end of the first part about Unitig safety. Now we move on to the second part where we talk about the palindrome splitting artifact. So for this, we recall our double-directed graph model here, and we can mark all the Unitigs from the double-directed graph. And similarly, we can do that for bidirected graph. We have this Unitigs labeled here. So notice that this particular Unitig TATA in double-directed graph, it is split into 2 halves in the bidirected graph, right? Like TATA is present as AT and TAT in this graph. And we also note that TAT is a palindromic Unitig. So it's we can kind of observe that when there is a palindromic Unitig, it's going to be split into 2 halves. And we proved that formally in this theorem 2, where we established the relationship between bidirected graph and double-directed graph maximal Unitigs. And as you can clearly see from the figure, there is a 1-to-1 correspondence between the non-palindromic maximal Unitigs in doubled graph and the red no isolated loop maximal Unitigs in bidirected Debrun graph. And when we look at every palindromic maximal Unitig in double graph, it corresponds to 2 maximal Unitigs in the bidirected Debrun graph here in this 2 green color. These are basically the 2 halves of the palindrome. So one is an isolated loop at start Unitig. So ATA, it has an isolated loop at its start. The other is TAT, it has an isolated loop at its end. Okay, so that's the theorem, and that confirms that palindromic splitting artifact is real. The isolated loops basically break the palindromic unitics in half, and this can cause underassembly. So that brings me to the end of our theoretical results, and we can move into the experimental results to confirm that these artifacts is present in real data. So first of all, we want to look at the unsafe and misassembled unitics. So recall that unsafe unitig means that those are not substring of the sequence segment. But unsafe unitics doesn't always translate to misassembled unitics, because misassembled unitics are basically the ones that are not substring of the reference sequence. It's possible that unitig is unsafe, but it's not misassembled. So we kind of have results for both of this in this table. We use the dataset T2T human chromosome 1, and we simulated error-free reads of length 100 of different coverages. And then we also calculated the unitics, which are the directed unitics using directed version of RECOMB. And we do see some number of unsafe and misassembled unitics. And not all the unsafe unitics translate to misassembled unitics, as we can see from here. The other thing to notice here is that as coverage increases, the number of misassembled and number of unsafe unitics go down. So since the coverages here are quite low compared to what modern sequencing technologies can do, we ask ourselves the question, is this artifact present when the sequencing depth is high? So to answer that, we had to use a metagenomic data set. We use CAMI low complexity data set, and we needed to resimulate it using the list of 30 genomes in the paper. And we use the same relative abundances and average covers that they have in the paper. And the reason we had to resimulate is because we want error-free reads. Okay, so we use reads of length 150 and K55 to get this 37 misassembled unitics. So even with high sequencing depth, we do get some misassembled unitics. Now we want to see if this like 37 misassembled unitics, do they actually appear in the final contig output of real assemblers? Because the assemblers do not only stop at the unitic stage, it will do some more, use some more algorithms to end up with the contigs. So are these artifacts present in those contigs? We see yes, they do. And we tested these two different assembler, megahead and metaspade, for metagenomic assembly of the CAMI dataset. And we see that out of those 37 misassembled unitics, most of them corresponds to some misassembled contig in both megahead and metaspades. Okay, so that was the first artifact. And how about the presence of the second artifact in practice? So first we ask, is this palindromic splitting artifact present in real genome? So we use this chromosome 21 and built unitics from it for different values of K. And we do see there are some numbers of palindromic unitics that are split, and it reduces as K increases. Now we want to see if it's present, if this artifact is present in the output of the assembler. So because the long palindromes are not very common in real genomes, we had to artificially introduce many thousand length palindromes in human chromosome 4. And from that, we simulated reads of length 100, definitely error-free, with coverage 10x. And then we also computed the palindromic unitics from them using K32. And then we want to categorize those palindromic unitics into this 3 category. So we say a palindromic unitic is fully covered if palindromic unitic W is fully covered if there is a contig whose alignment spans W's, and we say the palindromic unitic W is split if wherever there is a contig that goes through the middle of W, the contig is split. And the other categories of palindromic unitics that doesn't fall into either fully covered or spade is ambiguous. So for assembler megaheight and spades, we see all the palindromic unitics are most of them are fully covered and none of them are split. For Abyss and Minia, we do see some split palindromic unitics, so that does confirm that palindromic splitting occur in practice. Okay, so to summarize, our contribution is that we theoretically predicted the existence of assembler artifacts, and we showed that even without sequencing errors, misassemblies and underassemblies can occur, and we demonstrate their extent in practice. And we showed like it's kind of limited, and there are some possible fixes for the misassemblies. Like in case of whole genome assembly, you can increase the coverage to possibly fix the misassembly or break the unitics across uneven coverage. However, it's not so simple for metagenomic assembly. And there's also this possible fix for underassembly where you can force the unitics to push through those isolated loops. So that brings me to the end of my talk. And I'd like to thank everyone who participated in useful discussions about this project. I'd like to thank the funding sources. And last of all, I'd like to thank you all for listening to me. And I'll be happy to answer any questions that you may have. Thank you.